Introduction. Cell division, the process by which a single cell gives rise to two identical daughter cells, is a fundamental mechanism that underlies the growth, development, and reproduction of all living organisms. This intricate process involves a sequence of events that ensures the faithful replication and distribution of genetic material. In this article, we will explore the stages and significance of cell division, shedding light on the remarkable mechanism that perpetuates life's continuity. The cell cycle, an overview. The cell cycle is the series of events that cells undergo as they divide and replicate. It consists of interphase, which prepares the cell for division, and mitosis or meiosis, where the actual division occurs. Interphase. Interphase is the longest phase of the cell cycle, during which the cell carries out its normal functions and prepares for division. It can be divided into three stages: G1, Gap1, S, synthesis, and G2, Gap2. During S phase, DNA replication takes place, ensuring that each daughter cell receives a complete set of genetic material. Mitosis or meiosis. Mitosis is the process of cell division that generates two identical daughter cells, while meiosis is a special type of division that produces cells with half the number of chromosomes, essential for sexual reproduction. Both processes involve a series of distinct phases: prophase, metaphase, anaphase, and telophase. 2. Mitosis, the continuity of life. Mitosis is the primary form of cell division in somatic cells, ensuring the growth, repair, and maintenance of tissues and organs. Let's explore the stages of mitosis in detail. Prophase. During prophase, chromatin condenses into visible chromosomes, each consisting of two identical sister chromatids held together by a centromere. The nuclear envelope breaks down, and the centrosomes migrate to opposite poles of the cell, forming spindle fibers. Metaphase. In metaphase, the spindle fibers align the chromosomes along the equatorial plane of the cell. This alignment ensures that each daughter cell receives an equal number of chromosomes during division. Anaphase. During anaphase, the sister chromatids separate and are pulled toward the opposite poles of the cell by the shortening spindle fibers. Once separated, they are considered individual chromosomes. Telophase. In telophase, the chromosomes reach the opposite poles of the cell, and new nuclear envelopes form around each set of chromosomes. The chromosomes begin to decondense, and the spindle fibers disassemble. Cytokinesis. Cytokinesis, the final stage of cell division, involves the division of the cytoplasm and the formation of two distinct daughter cells. In animal cells, a cleavage furrow forms, constricting the cell membrane until the cell splits into two. In plant cells, a cell plate forms between the two daughter cells, eventually developing into a new cell wall. Conclusion. Cell division, whether through mitosis or meiosis, showcases the intricate machinery that perpetuates life's continuity. The seamless coordination of events ensures the faithful replication and distribution of genetic material, allowing organisms to grow, develop, and reproduce. Understanding the stages and significance of cell division not only deepens our appreciation for the complexity of life but also provides insights into the mechanisms that drive genetic diversity and the remarkable adaptability of living organisms. As we continue to unravel the mysteries of cell division, we gain further knowledge of the fundamental processes that underlie the wonders of life.